Okay, welcome back, and this is the last one for Chapter 18 and Unit 4. So this is going to go over development and organogenesis. So I've kind of mentioned some of this stuff before. You might hear some of these terms that you've heard before, but we're just going to go over them in a little bit more detail. So just a reminder, the egg and sperm are going to fuse to make the zygote. So as I mentioned before, you can only have the right number of chromosomes for it to actually work. Now there are protections out there to kind of prevent this from happening. You have this corona radiata and zona pellucida on the egg that are going to provide protection and nutrients to the cell. So you're just kind of looking at those guys right there. Now the acrosome of the sperm, which is also a part of the head, is going to have enzymes needed to fuse with and bind with the zona pellucida. And when that happens, the fusion of that the acrosome enzymes with the zona pellucida, what's going to do, it's going to trigger some reactions in the cell. And what that does is it causes proteins to release from the egg to actually prevent other sperm from fusing with it. So this is a video that kind of goes through that process, and it'll be a good review video from the last lecture too, talking about how it progresses through once it's ovulated down the fallopian tubes, and then it kind of hits on some of the things that we're going to talk about on the next slide too. So once the zygote is formed, it's going to undergo several rounds of rapid cell division. It's just going to keep undergoing mitosis over and over again and form this hollow ball of cells, which is called a blastula. Now, eventually, the blastula is arranged to form what we call a blastulus, and it's going to look like this right here. It's going to have two cell layers. It's going to have the outer layer, which is called the trophoblast, and these are going to help with implantation into the endometrium. It's also going to have this little group of cells right here called the inner cell mass, and this is what's going to go on to form the entire embryo. Now, this is where we have the embryonic stem cells. This is the holy grail of stem cell research. We'll talk about it in one of our discussion posts. There's videos on it, and like I said, if you ever want to talk about stem cell stuff, don't be afraid to ask, and I could talk hours on it. So here's a little video that kind of goes through this process and talks to you a little bit more about this whole stage here. Now, if you remember, I kind of mentioned this when I talked about the animal kingdom and how we classify different ones, and we have the different layers of germ cells, and the process is called gastrulation. So eventually, what you're going to see is we're going to have our cells develop into three germ layers, either the endoderm, mesoderm, or ectoderm. And depending which layer of cells, and it's all about position, will determine what type of organ they're going to form or the type of cells that they ultimately will be. Now, the process of gastrulation, and this is a cool video because you can actually see it happen on a little frog egg that's going through development, is you're going to start to see this infolding start to happen, this blastopore. And if you remember the protostome versus deuterostome, it determines if it's going to be the mouth first or the anus first. Now, once these germ cell layers have determined, then they go on further stuff to become differentiation via organogenesis. And that's where this one really wraps up. They don't go into too much detail, and I wanted to make sure I didn't go overwhelming with it too, because the last chapter has a lot of information. Now, what's going to happen, depending on location, it's all due to certain genes turning on at certain locations. Those germ layers will then develop into the different systems. Now, this process of differentiation all comes down to genes. It's location, certain things turning on and off at certain times. And it's all going to be causing all these different signaling cascades. And like I said, location and timing are key. And that's why we really love to understand why genes, how we know important they are for development. Several organisms are essential for studying this. The fruit fly, the worm C. elegans, and Xenopus the frog, they are key organisms used for developmental biology and understanding what genes turn on at what time, how they're important. I've mentioned Hox genes before, if you remember when I talked about them, especially in the evolution part. But these are genes on a chromosome, and we share a lot of similarity of these genes that you might see right here are pretty much almost identical to in humans, which is kind of cool. And it's, as I mentioned, as position. These genes have to turn on in the right location, because we've seen it as scientists, we can actually take maybe a gene that's essential for this leg, we move it up here, we can actually have a leg growing out the head of that fly. Like I said, this can get super complicated, but I just want to keep it kind of simple for y'all. Once again, some review videos and credit for the images. And that's it for this